Okay, so let's go through some of the nitty-gritty details of the salt and water handling in the nephron here. So here's a quick overview. In the proximal tubule, we get what's called isoosmotic reabsorption. And what that means is that the fluid, when it's filtered, has the same osmolarity as the plasma. And what's going to happen is, more or less, through the proximal tubule, that osmolarity will remain the same osmolarity as the plasma. And the reason is because you're transporting sodium out, water follows the solute, and so the osmolarity of the tubular fluid is maintained because you lose the same amount of water as you do solute. Okay? We'll talk about that in a second. And the loop of Henle, this is where we separate salt and water. The water reabsorption in the descending loop, the sodium reabsorption in the ascending loop. The distal tubule and the collecting duct, the distal tubule completely impermeate to water. Some sodium transport in the collecting duct. This is where our reabsorption is regulated, and this is where we determine through hormones, like antidiuretic hormone, whether we are going to get a dilute or concentrated urine. Okay? So here is a figure that I've already shown you with the sodium transport in the proximal tubule. This is a rather important graph describing what happens to the ion concentrations in that tubule as water moves. So let's take a look at this. We have distance along the proximal tubule on the x-axis. So this is the beginning of the tubule. This is toward the end of the tubule. Okay? And these are concentrations of ions as you go from the beginning to the end. So we have relative concentrations to the plasma of glucose and amino acids, bicarb, sodium, and chloride. Okay? So if we take a look at this right away, you can see we have sodium-dependent transport of things like glucose and amino acids, and those are going to be completely reabsorbed in the proximal tubule. We've already talked about that in a previous lesson. Okay, so the red line basically drops to 0 as you go from the beginning of the tubule to the end. It's basically gone by the time you've gone through the proximal convoluted tubule. All right, let's take a look at sodium. The sodium concentration doesn't vary. It's exactly the same concentration as the plasma at the beginning of the proximal tubule. It's exactly the same concentration as the plasma at the end of the proximal tubule. Okay. The reason is because as sodium is reabsorbed via mechanisms that I've already shown you, what you do is you locally, as the sodium is moved, and the chloride, as the sodium is moved, you create a higher osmolarity locally where the sodium is transported across. And so water will go from high osmolarity to low osmolarity. Now do bear in mind that we're not talking about a big gradient, okay? We actually call this isoosmotic volume reabsorption, and you can see we're saying that the sodium concentration doesn't change through the tubule. There is an osmotic gradient that's driving that water reabsorption, but it's very, very small. For all practical purposes, not measurable, okay? And so we can still call it isoosmotic. Don't let that confuse you, okay? All right, so the sodium concentration stays the same because we're moving sodium and we're moving the exact same amount of water to keep that concentration the same, okay? The chloride initially rises, and early it actually goes up. And this is because water is being transported out. It's following the sodium, but chloride is not. So chloride is becoming a little bit more concentrated as you lose water, but chloride's not moving. So I told you guys, when you move a cation, you have to move an anion with it. And I told you the majority of the time it's going to be chloride. So chloride parallel sodium. So what's the deal? Well, early in the process, early in the proximal tubule, the anion that follows sodium is bicarb, okay? So because bicarb is moving along with the sodium early in the proximal tubule, the chloride doesn't move. And so water goes out, chloride stays in the proximal tubule, and the concentration goes up, okay? All right. Later, along the proximal tubule, the chloride levels level off. And now this is because we've basically gotten bicarb so low that it's not being transported as much anymore, really hardly at all. And so because the bicarb is low, basically it's been transported out sort of farther along the tubule, now chloride starts to move and follow the sodium. And so chloride is now moving out along with the sodium and the water. And because it's moving proportionate to the water, the concentration doesn't change. And you get a leveling off of that concentration late. So it rises initially, but then later as you run out of bicarb and the chloride starts to leave the tubule along with the water, the concentration levels off and doesn't change, okay? Or not as much. All right. So moving on to the loop of Henle, again, this is where we separate our salt and our water. Note that you always reabsorb more sodium than water. So for instance here, we've reabsorbed 10% water here, the sodium transport, 25% reabsorption, okay? And that means that the fluid leaving this ascending loop is going to be relatively dilute. So the sodium and water reabsorption are physically separated by the descending loop and the ascending loop, as I said before, and that is obviously something to bear in mind. The sodium reabsorption in the ascending loop of the thick ascending limb of the loop of Henle takes place via the figure sort of represented here. First of all, recognize our old friend, the sodium-potassium ATPase drives it all by keeping the sodium concentration inside the cell low, okay? And that drives transport. The apical transport takes place through a sodium-potassium 2-chloride symport mechanism or co-transport, okay? So this transporter is the target for furosemide, which I bet most of you have heard of. If not, you will certainly get plenty of Lasix talk in pharmacology, but this is the target for Lasix. And so if you inhibit this transporter, you leave sodium in the tubular lumen, in the ascending loop, and that makes the osmolarity in the ascending loop and the distal convoluted tubule higher than usual. And because the osmolarity is higher than usual in the tubular fluid, you retain more water and you end up with a dilute urine, okay? You're leaving sodium behind, okay? Note that all three of these ions are required for this transport to take place. If you run out of potassium, it won't work. The sodium and chloride can still be there, it won't matter. Without potassium, it won't work. They all three need to be there. Potassium is, of these three ions, the scarcest, and so there is actually a potassium channel here, which allows potassium to come back out again into the tubular lumen, and that keeps potassium available for this transporter to work, okay? In the distal tubule, we have sodium reabsorption also by kind of a unique mechanism. Of course, our old friend, the sodium-potassium ATPase, is here, driving the transport of the sodium, keeping the intracellular sodium down, okay? And now, our transporter across the apical membrane, instead of being a sodium-potassium 2-chloride, is a simple sodium chloride symport or a simple sodium chloride co-transporter right here. So sodium and chloride go in, okay? That transporter is the target of thiazide diuretics, another biggie that you're going to hear from our pharmacologists, okay? You may note that there are also apical calcium channels here that are regulated by PTH, parathyroid hormone. We will talk about the details of that in a later renal lesson that will be coming up. Okay. And finally, in the collecting duct, we actually have two kinds of cells. Okay, we have the intercalated cells, or the type B cells, and these there are two subtypes, A and B. This is a type B cell. The intercalated cells are responsible for chloride and bicarb transport, so we're not going to talk a lot about that right now in this lesson, but bear in mind that it's there. And then what we are going to talk about are the principal cells, and these are the cells that are responsible for transporting sodium across the collecting duct epithelium. Okay, so again, our old friend, sodium-potassium pump, driving sodium transport, keeping the sodium concentration down in the cell. And yet another unique mode of transport. This time we have a sodium channel and the apical membrane. This is not the same sodium channel as the fast sodium channel that we've talked about when we talked about action potentials. It is not voltage dependent. It is regulated by aldosterone and not things that commonly inhibit fast sodium channels like tetrodotoxin. Okay, so this is a unique channel that is here, and it allows sodium to go from high concentration to low concentration, and again, the low concentration maintained by the sodium-potassium ATPase. We will talk about aldosterone in a later lesson, but for right now, be aware that this channel is regulated by aldosterone, and it is also the target of some diuretics that you will talk about probably quite a bit more with me and with the pharmacologists. Okay? All right. So here we have another view of the principal cell. This time we've added aquaporin, or water channels, that are under the control of antidiuretic hormone. Okay? So the permeability of the collecting duct to water is inherently very low. It is not zero, but it is low. But if you add antidiuretic hormone, that increases the permeability. And the reason it does that is because it takes these water channels, these aquaporins, which are usually in vesicles, and it causes them through a form of exocytosis. It causes these vesicles to merge with the apical membrane so that these channels are now inserted into the membrane, and that increases the water permeability. Okay? ADH released from the posterior pituitary acts on the apical membrane of the principal cells, increases water channels, increases water permeability. All right? So some important points to remember. The collecting duct is the only site where the water permeability is hormonally regulated. Okay, and ADH increases that permeability. Second, the collecting duct does have some water permeability, particularly deep, particularly in the intermittent medulla, I guess, in the absence of ADH. So water will always be reabsorbed. It just won't be reabsorbed very much. Something to bear in mind. It is a dose-dependent action. In other words, high ADH will give you high permeability, low ADH will give you low permeability, a medium amount of ADH will give you medium permeability. Okay, it isn't like all or none where all the channels go in or out depending upon whether there's ADH present. It's a dose-dependent response, and I already said this, it stimulates this permeability by causing aquaporin to be inserted. Bear in mind that when ADH drops, that aquaporin channels go back into vesicular form. So it actually is a reversible process where ADH might go down and these water channels are suddenly brought back into the cell via endocytosis and removed from the apical membrane. So it is sort of an up and down thing. If there's no ADH present, you are going to not have water reabsorbed from the tubular fluid, and you will have a dilute urine. If there is a lot of ADH present, then you're going to have a high water permeability in the collecting duct. You're going to allow water to be reabsorbed into the inner medulla, which has a very high osmolarity, and you will have a concentrated urine. Okay. All right. Let's cut that off there. And the last thing we'll talk about is what maintains that really high osmolarity in the renal medulla.